Welcome to the Social Media and Science Communication Mini Symposium organized by Eva Hellman from the Sainsbury Lab at Cambridge University in the UK and myself, Javier Brumos from the Institute for Plant, Molecular and Cell Biology from the CSIC in Valencia, Spain. So why did we organize a session about social media? Why is social media so powerful? So it has transformed the way we communicate. In social media, when we observe the communication process, the sender and receiver roles are totally dynamic. And what I mean is that the content is created or co-created by the platform users. The users interact and discuss the content with others, and anyone can participate. It's a universal audience. And who is this audience? So in the latest statistics that we've seen, there are more than 4.6 billion daily active social media users, which is about 59% of the current world population. And if we break this down by age, we see that younger generations, at least in the example of the US, are using social media more often, but older folks are also pretty much engaged in social media. And how much time do we spend in social media? So it's an average of 2.5 hours per day per person we spend in social media. And when you ask these people, why do we use social media? There are two main answers. One, to keep in touch with friends and relatives, and also to stay informed on current news. So social media has transformed the way we consume and access news. Audiences turn to social media looking for information. And the problem is that we don't have any quality control. Nobody needs to be an expert to express their views out to the world. For those reasons, scientists should be at the forefront of social media engagement. We can provide data-based facts to an audience that is eager for information they can trust. For those reasons, we have an unbeatable opportunity as scientists to take the stage and contribute to the education of millions of people. So as scientists, we can use social media, and we have invited several speakers today that are going to be presenting their work and go over this topic. So as scientists, we can make science more approachable and engaging for the general audience using social media. We can share this science with the public, with policymakers and other scientists. We know that educated public can affect and understand policymakers and legislators' decisions. We can disseminate scientific discoveries and keep up with recent publications. And this contribute as well to the sense of community, to the sense of belonging, to be part of something and networking. So these are the different seminars that we have today in our mini symposium. Mary Williams is going to be talking about the American Society of Plant Biologists' social media journey to support, amplify, nurture, and connect plant scientists. Cezanne Daniel is going to be talking about science communication and community building as an early career researcher. Detlef Weigel is going to be talking about social media and public engagement with the wider community and policymakers. And Elizabeth Haswell is going to be talking about social media to build and broaden the plant biology community. After these seminars, we will have a live panel discussion together with Veronda Montgomery, and we will address the submitted questions. Please submit questions during the seminars, and especially you, the PhD students and the early career researchers. So Mary is going to be talking about social media and science communication from a society's perspective, the ASPB. Her work over from biochemistry to plant molecular biology. After 14 years as a professor, she became part of the ASPB, and she has developed the teaching tools in plant biology and is the features editor of the Plant Cell. She's involved in education and outreach, leading workshops on career, teaching, writing for PhD students and postdocs. And she's a Plante fellow involved in teaching the assistant features editors plant physiology. So Sesen will talk about science communication and community building. As an early career researcher, she's interested in plant genetics, especially underutilized species. She's a PhD student at the Cold Spring Harbor Laboratory, and her outreach work focus on debunking fake news about plant breeding, network building, social engagement and integration, and promoting intercultural exchange. She's a co-founder of the Progetto Bio Tecnologico outreach program and coordinates the ASPB African Researchers Network. She's an ASPB Convivium Scholar. Detlef Weigel will be talking about social media and public engagement with the wider community policymakers. His work covers from developmental biology to evolution. He is a director of the Max Planck Institute for Developmental Biology, and he is engaging in social media with policymakers about GMO regulations, and this sometimes leads to not so much joy with the lobby groups. He's also famous for the lab-related music videos and outreach activities. He is on various committees, societies, advisory and editorial board. He's in eLife, and he's a co-founder of 3 startups. Elizabeth Haspel will be talking to us about social media and building the research community. Her work covers from biochemistry to mechanobiology, and she works on these mechano responses in plants. She's a professor of biology at Washington University in St. Louis. And her mission statements include science communication, community building, mentoring, and teaching. And she will be talking about the podcast that she hosts, the Taproot, on Plantae with Ivan Baxter. And later in the live discussion panel, Veronda Montgomery will join us. Her social media focus on fostering equity and inclusion, the increase of visibility of Black researchers. Her work is on photobiology and the adaptation of photosystems. She's a professor of biochemistry and molecular biology at Michigan State University. She's a science defender, writer, science communicator, AAAS fellow connecting science and policymaker. She's a co-founder and organizer of First Black Botanist Week. She's an assistant vice president of the Office of Research and Innovation. And please read her new book, Lessons from Plants. So remember to write your questions for the panel discussion. And with that, I will leave it for Mary to start her presentation. Thank you so much.
Hi, thanks for inviting me to speak in this session. My name is Mary Williams. You can find me at mwilliams@aspb.org, and on Twitter, I'm @PlantTeaching. I'd like to talk to you today about ASPB's social media journey to support, amplify, nurture, and connect plant scientists, and my journey also. The ASPB is the American Society of Plant Biologists. It's nearly 100 years old. It was founded in 1924. It's a professional society for plant biologists. You may be familiar with our journals. We publish the Plant Cell and Plant Physiology and co-publish Plant Direct Journal. We're the founder of the Plante.org website and community site. We also host several meetings every year. The annual meeting is the plant biology meeting. We also host regional and topical meetings. So if you're not familiar with this, please look us up and join us. So we were asked to talk about our journeys through social media, and that prompted me to look into the archives, and I found my first tweet, which was nearly 10 years ago. And I was at a conference on science policy and science communication. So I tweeted, here is an article that tells us why we need plant scientists and plant science educators. And I linked to an article in PNAS called Global Food Demand and the Sustainable Intensification of Agriculture. I'd like to thank two people who started me on my journey, and these are Sarah Blackford and Ruth Bastow from Society for Experimental Biology, Garnet, and Global Plant Council. They convinced me that Twitter was a good way to share the things I was working on, and they were, again, at the start of my journey. Thank you very much for that. Why I started was because I'd been hired in 2009 to develop the educational series Teaching Tools in Plant Biology. That's a feature of the plant cell. And as we started posting these on the journal website, I realized we didn't really have a good way of communicating about them to people. So thanks to Ruth and Sarah's nudging, I started these accounts. I started a Twitter account, a Facebook account, also a Scoop It account, which is sort of like a Pinterest bulletin board thing, which I don't use anymore, but it was great when I was using it. And I've got some more information on my poster, but we did a little study and found that really it was important to use both Facebook and Twitter to reach the audience we wanted to reach. Part of that is because Facebook has a much more international reach than Twitter does. And please, visit my poster and I can tell you more about that. I also wanted to point out that if you're just getting started on Twitter and you don't have a lot of followers, don't worry, it takes time to build up, but it'll happen. So that slide was from a 2012 presentation I made to the Plant Cell editorial board. And I was trying to convince the editors of the Plant Cell that social media was a good tool that we could use to communicate with authors and prospective authors about what we do at ASPB. And it was a bit difficult because some of them had this idea that social media was really just about angry cat memes and celebrity gossip and sort of something for kids. But I convinced them that it is in fact a legitimate and important way that we can share information. And I talked to them about the fact that just like in biology, simple signal amplification is very important. So we might put out one message, but other people will share and amplify that message. And so that's different than, for example, traditional media, where you might put out an ad or an article and it just sits there. Social media is a social network. So people share things amongst themselves, which is a very powerful part of social media. We established some goals, which were to raise the profile of the journal, to support the authors, and to provide the opportunity for dialogue. So this is another reason it's different than traditional media, is that it's a two-way street, and we can put out information and then hear how other people respond to the information we've sent out, which is very important in terms of helping us to direct what we do as a professional society. Here's an example of some of the ways we use it. But all of our journals put out a tweet and a Facebook post of every article with a link to the article to help promote the information that we publish. We also invite first authors to send us their profile, their little biography, and their picture, and we share these through the journal channels. It's a nice way of thanking them and congratulating them on getting their work published, but also it's a little hard sometimes to say, hey, look at me, look what I did. But if it's simply a matter of sharing something the journal's already done, it maybe is a little bit easier to sort of say, hey, look at me, I got a plant cell paper. We also share information. For example, we have a 2022 focus issue on plant-biotic interactions coming out. So I want to thank Linda Palmer and Suzanne Cholwick, who are in our subscriptions group, and they do the work of posting these things. Although it's free to use Twitter and Facebook, it does take time to format the posts and things. So it is something we're committed to doing, again, to support our authors. Another reason that we use these tools is because altmetrics is an alternative metrics. It's another way of measuring the impact of an article. And before an article has time to start accruing citations, you can see that the article is getting attention through this altmetric score. And the score is you can see a table that sums where all these different bits add up to make the altmetric score. And Twitter is actually very important because Twitter is public. Every time someone shares a link to this article, it adds to the altmetric score. So if you're not already using Twitter, but you're considering it, that's, I think, a good reason to at least join Twitter and start to amplify your work and your friends' work. Here's a tip. I've got more tips on my poster, but if in fact you are using multiple accounts or multiple platforms like we do, there are some tools that can make it a bit easier. For example, we use Buffer that allows us to post to LinkedIn, Twitter, and Facebook all at the same time. We also use TweetDeck, which allows us to tweet from multiple accounts with a single sign-in. And Katie Rogers is our community engagement administrator. She joined us a few years ago as a social media intern, but she's got a real skill at this. And so she's now in charge of a lot of the strategy and analytics we do to help guide how we use these tools. So if you have any questions about how to use social media in a sort of organizational capacity like this, I really recommend you get in touch with Katie. And you probably also know Katie, who's the host of our Plante Presents and Plante webinar series. Speaking of which, we have some recorded webinars that you might be interested in to learn more. One is called Social Media for Scientists, What, Why, and How. And the other is How Top Scientists Use Twitter to Support Their Research. So look on the Plante webinar, the Plante website, or contact me if you'd like direct links to these, but they're full of really useful information. Okay, so now we are where I am in my journey today. And we'll start with how I got interested in science in the first place. So I was a young person, was trying to figure out what I wanted to do with my life. And I discovered this great character called Mr. Spock from Star Trek. And it was immediately clear that that was who I wanted to be. I wanted to be able to look at evidence, make decisions completely based on logic without any complications like emotions or things like that. And I had this kind of dream that, you know, in a perfect world, we could all just be essentially brains under a dome, right? And we could all contribute whatever our brain, our ideas were. And, you know, that would, working together, we would, you know, make the world a better place. But of course, this is a very naive way of thinking. And it also, I think, reveals sort of the privilege that I came from, in that I was able to see myself as someone who could contribute based upon the ideas in my head. And you know, now I recognize that this is not true, right? It's not a level playing field. People have different abilities to access the structures of science. And that's where I am now in my understanding and thinking, is trying to think of what can we do to make science available to everyone who wants to participate. And of course, there are lots of reasons why it's inequitable. And this is a really nice image I got from New Scientist, where some of us see sort of blue skies, other people see barriers. So some of the factors that can influence whether or not we're able to contribute to science include access. You know, where are you born? Do you have access to a high school science teacher? Can you get into a university? You know, can you take the college prep classes you need to take? If you're at university, can you get access to maybe a research opportunity that will help you move into graduate school? And even once you get access and into science, then different people face different levels of discrimination and bias. There are microaggressions that can interfere with people's ability to contribute. Financial resources are another big issue. For example, can you afford to buy the laptop you need to do your work? Can you afford a subscription to a journal, or the fees to attend a conference? Do you have the financial resources to perhaps get childcare if you get invited to attend a conference? You know, can you? And then, of course, there's all sorts of access of ability and accessibility and accommodations. You know, are you able to listen to this webinar? Yes, indeed, because it will be captioned. So I'd also like to thank two of my friends who have really inspired me and helping me to learn to think about this part of my journey. And these are David Asai and Veranda Montgomery. Two real, you know, leaders, I think, in understanding how science is not a level playing field. We all have different challenges to overcome to participate. One of the things that David says a lot is we need to learn to listen to hear. And that is something that really resonates with me, because it's easy to talk. It's much harder to listen, and it's much harder to listen to hear. And so again, that's where I am now. I'm learning to listen to hear. I'm trying to learn and understand what ASPB and I and others can do to help allow everyone to contribute to science, to the best of their ability. So I want to give a shout out to some of the people whose voices I'm currently listening to very hard right now. Asia Hightower, Edith Pierre-Jerome, and Imani Madison are three people who put together a bunch of resources, including the Front and Center Plant Science Instagram page. So if you use Instagram, I recommend you follow it. Some other voices are the LGBTQ+ plant scientists on Twitter, Timber Burnett, who's calling out the fact that there's only 2 genders on some of the forums that we use, Diversify Plant Sci, which curates a list of individuals you might invite to give a seminar. Black Botanists, of course, introduced us to a lot of different people. Disabled in STEM raises awareness of some of these issues of accessibility. And so finally, I just want to mention a couple of resources we've developed. Changing Cultures and Climate is a web page on Planté where you can find lots of information. And ASPB Forward is a new committee, again, focused and targeted with addressing the inequities and trying to resolve some of them so that we can all participate. I look forward to talking to you, and I also hope that you can meet me at my poster or connect with me on Twitter. So thank you very much, and I look forward to learning more from you. Thank you.
Hello, everyone. It is really a great honor to take part to this community session and to speak alongside an amazing lineup of speakers, many of whom I looked up to myself in my science communication efforts. So today I will bring my perspectives as a graduate student and as an early career researcher who has started out in science communication and advocacy alongside her research. And I will try to explain why I got interested in science communication, how I do science communication and what I've learned from it, and why I think there is a need for diverse voices in SciComm. So let's start with the first question. Why did I even get in SciComm in the first place? And let's make a small premise here. So by the time I started my master's in agricultural sciences and molecular biotechnology in Italy in 2018, the European Court of Justice had ruled that precision breeding using genome editing was subject to the 2001 EU directive regulating genetically modified organisms. And just to give a bit of a context to folks that are not very familiar with this, the 2001 EU directive imposes strict legal regimes on the release of GMOs in the environment based on the so-called precautionary principle. And in practical terms, with this 2018 ruling, crops obtained through genome editing are regulated as crops obtained through transgenic techniques, establishing a sort of moratorium on these techniques that makes it almost impossible for the application of research in the field. And of course, these policy decisions startled many European plant scientists and also confused a young master's student like me who was just embarking on the journey and who was starting to realize that science was moving faster than regulation and governments, and who was deeply concerned that the polarized debate and uncertainty in regulatory frameworks would ostracize genome editing the same way it did for GMOs. And for my generation, that means missing out on opportunities to address critical challenges of our time. And we as a scientific community cannot afford that. So that's what got me in science communication initiatives such as Progetto Bio Tecnologico, an Italian initiative that I coordinated together with colleagues from the 3 universities in Pisa, and in a science outreach initiative that I coordinated together with American Society of Plant Biologists, ASPB Scholars. So let's start with Progetto Bio Tecnologico. Progetto Bio Tecnologico really started as a way to share our enthusiasm about biotechnology and make the debate on genome editing and genetic engineering more accessible to the general public. And we tried to do that by using dialogue-based forms of science communication involving students and early career researchers and aimed to other students. So the main targets of our initiatives were students in particular, because we believed that promoting scientific and critical thinking on highly debated topics at that stage would allow students to become advocates in their own communities and perhaps also motivate their career choices in biotechnology. So we were very active on social media, and in particular on Instagram, which is the platform mainly used by our audience. We took part to the European Biotech Week by organizing a public screening of the Food Evolution movie and organizing webinars. We also organized virtual lectures in local Italian high schools. And during the lockdown months, we became actively involved in debunking fake news on the pandemic and raising awareness on the importance of vaccines by making videos like this one with the "I will get vaccinated" tag. And more recently, we ventured in writing, and we also wrote in local Italian newspapers and in Italian SciComm journals. So while all these activities proved very, very rewarding, the backlashes received for our pro-biotech agenda made us realize that the deficit model, which assumes that a more scientific literacy and more information is all that people need to induce positive attitudes and feelings of trust towards science, and that it is all that people need to make better informed decisions, simply didn't work. And we also realized that the process of debunking fake news was perhaps less efficient than we thought, because of course misinformation traveled faster than debunkers themselves. And so we realized that we needed to learn how to frame information. We needed to learn how to place scientific information within an interpretative narrative and context before it could serve as a starting point for discussion and action. And in order to do this, we turned out to great mentors, including Professor Pamela Ronald, Professor Peggy Lemo, and Professor Alison Vaninanum, and great Italian science communicators to teach us the complex dynamics of science communication and the cognitive biases that communicators often have to face. And these interactions and the initiative in general provided us with 3 key insights that I would like to share with you. So first, communicating science, especially when it comes to highly debated topics such as genome editing and genetic engineering, and especially in agrobiotech skeptic context, can be very hard because everyone has different backgrounds and brings different understanding and culture and ideological positions to the table. And so trying to communicate without a common ground is not feasible. So what we learned was that the first thing that we needed to do as SciCommers was listening, something that we should also do in all of our scientific endeavors, by the way. And this means getting to know our targets, getting to know our audience's understanding, concerns, interests, and tailoring our initiatives and social media engagements to our audiences. For instance, we understood that high school students were less keen on reading an 800 words scientific blog, which was easier to write for us, but were much more likely to read an Instagram post organized in 5 to 8 slides of about 30 to 50 words each. Now, imagine how complicated it is to reduce a complex biological concept in an Instagram post of about 5 to 8 slides of 30 to 50 words each. And this brings us to the second point, which is simplifying without trivializing, also known as the power of storytelling. So each concept can be explained using a couple of sentences or with an entire book. And to simplify a concept, it is perhaps less useful to describe large amounts of data and experiments, whereas it is much more powerful to tell a story that fascinates and involves. And this is the power of storytelling. And the third insight is the importance of mentorship. So communicating science as a student and as an early career researcher can be very hard. And speaking up, getting exposed, and eventually also facing the backlashes can be excruciating. But that's where it becomes essential to have a network of peers and mentors from which to learn and seek support. Let's now turn to another outreach initiative that I coordinated with fellow ASPB Scholars, Modesta Bugu, Godwin James, and Aya Bridgeland. So the outreach initiative involved about 150 students from a couple of schools in Florida and aimed to measure how the knowledge and attitudes towards biotechnology changed after a workshop on genetic engineering in agriculture. And what we understood was that this cross-age peer teaching approach contributed to induce more positive attitudes towards biotechnology and also a greater propensity in students to act as advocates in their own communities. And we are now trying to extend this cross-age peer teaching approach in an international context. And before concluding, I would like to discuss another crucial point, which is the need of diverse voices in science communication. So most of the challenges we as plant scientists and science communicators seek to address in our efforts directly impact underrepresented and marginalized communities. Think about sustainable agriculture, about crop improvement, or natural solutions to climate change, or even vaccines, access to health and public policy. And quoting this great article by Professor Asmeret Assefa-Berhe, the absence of voices of marginalized communities in science communication and in public policy means that the issues that we are concerned about are not getting the attention they need. So let that sink in for a moment, and we might discuss it further in the discussion session. So I would like to conclude by saying that students and early career researchers can really contribute to efficient information framing by somehow bridging the gaps between expert panels and the general public and facilitating open dialogue and community engagement. And this is why we as a scientific community need to give ECRs a chance also in SciComm. Thank you, everyone, for your attention. I would really like to thank everyone who has supported me in this, in my SciComm endeavors. First and foremost, my team at Progetto Bio Tecnologico. Here is a picture with just some of the members in PISA. I would also like to thank the ASPB Scholars program and the fellows, and of course, the mentors that have supported me throughout this journey. Never forget the mentors. Thank you to the organizers for setting up this community session, and I would be more than happy to take your questions and start the discussion.
Hello, good morning, good day, good afternoon, good evening, wherever you might be. I'm really happy to be part of this mini symposium at ICAR 2021 on social media and science communication. Thank you very much, Eva and Javier, for organizing it and inviting me to this mini symposium. My name is Detlef Weigel from the Max Planck Institute for Developmental Biology. I tweet as Plant Evolution. Before we get going, a few disclaimers. In addition to my day job, I'm also being paid as a deputy editor at eLife. I consult for KWS. I serve on the medical advisory board of the Howard Hughes Medical Institute, and I'm a co-founder of a small biotech startup. So Eva and Javier were very kind, and they suggested a series of questions to cover to get the conversation later on with Mary Liz and Sesson going. And so the first question is, what platform and social media do we use? And I'm sure you all know this, that there is a very good number of platforms one can choose from. And some of these require good video material or work best if you have fancy photography. I found that Twitter works particularly well for us. We are also present in social media on Facebook, but that's mostly to communicate with current and former lab members, to share photos from soccer events or our holiday video, or somebody has been recognized in some way. So in addition to talks, seminars, and our papers, and of course our lab website, which we try to keep fairly well up to date, has been very challenging in the last year. The main channel with which we communicate very regularly with the larger world is through Twitter. And if I say we, it's really me who does the tweeting. If I have asked us that we should say something about why we started on social media in the first place, I have to admit this was 2009. A bit difficult for me to remember. I do know that Sofian Kamoun was an inspiration because he already had a very good presence on Twitter, a good friend and colleague from Norwich. And I have to admit that I first thought about this mostly in terms of communicating about things from the lab. But over the time, I've actually found that for me, perhaps the most important thing on Twitter is that it's a great channel to find interesting papers. And conversely, of course, I also comment myself on interesting papers. In addition, of course, I advertise our work, I advertise our people, I comment on science policy at large, then I try to engage with politicians and others on GMOs, gene editing, ag policy. That's again something that I hadn't really foreseen when I started on Twitter. So I like the format of Twitter. Back then, tweets were 140 characters. Now it's 280 characters plus URLs plus images. So you can say actually quite a bit. And you can say quite a bit, especially when you use threads. And this is actually an aspect of discovering papers that I really like. So you might have seen in one of the previous slides that I follow about 400 accounts or so. Others might follow thousands of accounts, but even with 400, 500 accounts or so, unless you spend an inordinate amount of time every day on Twitter, you can't see everything in your feed. So whatever you see is sort of a random selection, so that adds to this randomness of what you discover. And as I said, I really love it because I come very regularly across papers that normally I wouldn't see. So in the old days, I would go to the libraries, and the not quite so old days, I would get tables of contents emailed to me or RSS feeds, still get a few of those, but it will be mostly looking up things that are sort of in the area that I work on, interested in anyway. And so on Twitter, again, I love that I see stuff that I normally wouldn't see, and this is just one relatively recent example from Rotem Sorik's lab on bacterial immunity, which is really super cool. And so this tweet here, of course, very short, doesn't say a whole lot, although there's already this image here that you can look at. But then it's a whole thread, and thread is just a chain of tweets where you can explain what you saw there. So discovering work of others, I think that's really cool. And then similarly, I will comment on papers that I find particularly interesting, sometimes also when I have questions about papers, or rarely when they're papers where I'm a little bit suspicious that some things maybe don't quite add up. But overwhelmingly, it's really positive things. And I try to inject something personal often into this. So for example, here, wonderful paper from my colleague Ksenia Krasileva. And I thought it was really super cool. And I have to admit, I was a little bit jealous when I saw it published because it was so cool. And I thought, man, why did I not think of doing this? And I put it in the tweet, as you see. So I, of course, also use Twitter to advertise our work. So here, again, relatively recent tweet from something about something where I still think it's probably the most provocative study ever from my lab. So it's something that is not easily explained just in 280 characters, and this is where we resort to a thread. So this chain of tweets, and always here stage and image, and then have some comment on this. So it's whole chain. I spare you going through the entire thing, and then again I try to use something more personal to make comments. So here, for example, it's this work that seemed really pretty controversial, but I think we resolved it quite well. That in the end, a straightforward explanation. So now you're talking. And again, also shout outs to people in the lab. So here, Effie, when she graduated half ago, and you can see in the time where we had not to wear a mask yet. Also things more serious. Last year, I think all of us were giving a lot of pause of what was happening, mostly in the US, but not only in the US, things related to Black Lives Matter. And Bridget Waitaka, graduate student researcher in the lab from Africa, who has studied and lived in the United States. Bridget and I, we had some pre-series conversation, and I made a video of one of these conversations on diversity, tokenism, racism in Germany and other places. This is on YouTube, and again, we used Twitter to alert the wider world to this in case they wanted to watch this and hear what we would have to say. Related to this, Eva and Javier also asked us to mention whether diversity, equity, inclusion, DEI, whether that played a role in social media for us. I certainly took to heart what I read last year. I follow now a much more diverse group of accounts than I did a year ago. And I have to say that I really have learned a ton from following many of these accounts. So can only encourage you to look out and look out for people who you might not easily come across otherwise. I do also use Twitter to comment on science policy and science affairs. I try to stay away from party politics, but when it concerns science, I do comment on it quite regularly. Again, here's an example from last year. I had come across this article here in Times Higher Education Supplement. It was about a fraction of first earthers that were female over the years from 2015 to 2020. And they had this graphic here. So you see every year this was going up. But then all of a sudden in March of 2020, that trend was broken, even more so in April. And then May, it was very clear that the fraction of female first authors was going down substantially. And this is from thousands and thousands of papers. So this is highly significant, even though the scale here does not start at 0. Now, this is an impressive graphic, but when you first look at it, it might not be immediately apparent. And so I made a quick sketch here to make it more obvious what was going on, and then comment on it. And so you can see that a lot of people responded to this and made comments on this. Yeah, and then finally, this is something that I only gradually got into, that I've been engaging in discussions on GMOs, and especially now genome edits, and also ed policy at large more and more. I mostly do that in German, and these discussions with NGOs, but also with politicians, both from the federal parliament and the EU Commission. Of course, these are very short arguments, and I don't pretend that my comments will likely convince those directly on the other side of my viewpoints, but this actually does serve a larger purpose because this is being seen by bystanders and also being seen by journalists. So from this, I've had quite a few journalists contact me and ask me to comment on things that then appeared in articles or blogs, or that led to radio interviews or other forms of publicity on topics that I really care about. And that's really all I had to say. Thank you for watching this and listening to me. And again, I'm really looking forward to the discussion with Mary, Sesson, and Liz on this topic, social media and science. Thank you very much.
Hello, everyone. I'm Liz Haswell, and I'm excited to share with you some thoughts I have about using social media to build and broaden the plant biology community. I see this as one type of science communication. Outreach, as you've been hearing, focuses on communicating to those outside the scientific community by increasing a public appreciation of science, public knowledge and understanding of science, and influencing opinions, policy preferences, or behavior. InReach, on the other hand, is focused on communicating to members of the scientific community. This can include sharing your recent findings and excitement for science, and recruitment, retention, and community building. It's this last point, community building, that I want to discuss today. There are, of course, many methods for community building, but today I want to discuss a method near and dear to my own heart, podcasting. A podcast is essentially an audio magazine with regularly, or ideally regularly, spaced episodes that can be stored and listened to at the listener's convenience. Podcasting started in the 1980s, but really gained steam in the mid-2000s when the term was first used and when iTunes released their podcast app. Now, there are dozens of podcast listening apps and billions of podcast listeners. And podcasts have become an enormously successful medium. A survey by the Pew Foundation in 2018 found that 44% of people in the United States over the age of 12 had already listened to at least one podcast, and online resources suggest that this number has increased by 2021. And those listeners have a lot to listen to. There are currently over 2 million active shows. However, and this is my point, a survey in 2018 identified only about 1,000 active science podcasts in the English language. Of course, there are many others in other languages, and surely the number has grown since 2018. But still, science-related podcasts are just a sliver of the content out there. So I would argue that we are really missing a huge opportunity out there. And not just because so many people listen to podcasts, but also because these listeners are so diverse. On the right, you can see the stats for the self-reported identity of podcast listeners from 2010 to 2016. While again, these numbers are for the United States, so may not hold in other countries, the audience for podcasts mirrors US Census statistics. There are lots of other advantages to using podcasting for outreach and for inreach. The access is high and barriers to access are low. The barriers to production and distribution are very low. Production and distribution are decentralized, so you don't have to go through anybody else. If you want to put your podcast together and put it out there on iTunes, it's completely free to you. It's also a form of direct communication with an audience without going through a journalist. And it's an amazing way to reach geographically, demographically, or ideologically isolated individuals. If you have listened to just a few science podcasts, you know that they can look and sound a lot of different ways, and there's so much room for innovation in this space. Your audience could be other scientists, the public, children, or students. The goals can be very diverse. They can be research related, sharing and discussing science within the science community, communicating results to the public, can be social justice oriented, providing information, resource, and community, advocating for changes in policy, environment, or attitudes, and democratizing and disseminating career-specific information. And you can do this in a number of styles. There's the opportunity to give a monologue, to have an informal chat, to have more formal interviews or panel discussions, to give a highly formal lecture, or to give dramatic or comedic readings or skits. At the moment, most science podcasts are in the area of general science, as you can see in the panel here. There are some great plant biology podcasts out there, and they must be included in the biology section. Most science podcasts, as you can see here, are aimed at the general public and are associated with outreach, although there is a subset, about 16%, that's aimed at scientists. If you're looking for a niche to fill, it looks like science podcasts for children is an area with a lot of opportunity. But regardless of the topic or the audience, one thing you will find in common among most, if not all of these podcasts, is that they use the power of narrative to get their message across. It has been documented that people learn more easily when information is provided to them as part of a narrative, with a beginning, a middle, and an end, rather than a list of facts as you would find in a textbook. And there are some behavioral ecology rationalizations as to why this might be the case. So using the power of narrative is the approach that Ivan Baxter and I took in developing our podcast called The Taproot. This podcast focuses on telling the story behind the science as a form of community building. Ivan and I founded this podcast with the help of ASPB and Plante way back in 2017. Typically, each episode starts with a different primary research paper. We start by discussing the paper with one of its authors, but then quickly move on to the story behind that science. Because while a scientific publication tells a story about molecules, cells, processes, or mechanisms, but it doesn't tell all of the other stories that went into the data-driven story. Stories about how funding was obtained, how relationships between the co-authors were maintained, all of the experiments that didn't work, any time that technology had to be developed in order to get the experiments to work. All of those are stories that we try to tell in our podcast. And so the Taproot was born. Here's just a brief outline of how it happened. Ivan and I discussed starting a podcast while I was on sabbatical in Cambridge and New Zealand in 2016. We both were and still are big podcast consumers, and we wondered if we couldn't also be podcast creators. We pitched the idea, including the stories behind the science aspect, to ASPB and Plante in early 2017, and they agreed to let us give it a shot. From the very start, Plante provided editing, help building the website, financial support, did a lot of the advertising, gave us access to Mary Williams, and created our icon. Since then, we've released 5 seasons, starting in the summer of 2017 and just wrapping up this last February. Now, our goals and our intended audience help drive the content and keep us focused on what matters. Our intended audience is young plant scientists, and we have a number of goals: to normalize challenge and struggle, to expose young people and early career researchers to the real world of science, in particular, to provide access to the hidden rules of academia, and to help provide some examples of how to navigate careers. Here you can see the faces of our wonderful guests from season 5. In the off chance that this presentation caught your imagination and you're intrigued by the possibility of starting your own podcast, here are a few pieces of advice. Definitely start with a vision, where you want to go and what your goal is in putting a podcast together. Come up with a structure both for the season and for each episode before you start. Gather advice for good sound equipment, editing approaches, and so on. Don't get those from me, I don't know. Do a practice episode that will help you figure out what works and what doesn't. But really, the best advice I can give you is just to jump in and give it a try. I have found podcasting to be incredibly fun and rewarding. And with that, I will wrap up. I am very grateful to have had the chance to work with and be inspired by all of the people listed on this slide, and to have had financial support from Plante, ASPB, and NSF. Thank you for listening to my presentation, and I'm really looking forward to our panel discussion.